So today our topic is Volcano plot. We'll be discussing about the importance of Volcano plot, where and how we use it, how it explains our data, and what are the benefits it's providing. Then we'll go through the basic foundations of the Volcano plot a little bit, and eventually we'll go for the live demonstration, which is probably everyone prefer. So let's begin. So one of the basic purpose of using the Volcano plot is the visualization of our data. And to do so, we have multiple plots definitely, but Volcano plots give some benefits, exclusive benefits. When it started then, it was basically started by mathematicians a long time ago, but the adaptation with the biology and explaining the biological data with this Volcano plot started around 2000. So it basically speaks about two things. One is the significance and magnitude of change in genomics experiment. As the major purpose of our tutorial is to build up the bioinformaticians, so we'll concentrate mostly explanation that's relevant to the bioinformatics work. So this is one of the figure. That's this paper by Gary Churchill and Jenkins Kui was published in 2003. And if you look at this figure, the A log full versus locked in p-value and F1 change in here. It's in 2003, one of the earliest paper which was using the Volcano plot for explaining the data. Then we also use the first discovery rate, which is also around 2003. Anna Trainer and Yuval Benjamini was using from the University of Tel Aviv. First discovery rate we extensively use on different purposes, including the bulk RNA-Seq. So let's go back to the basics. What is Volcano plot's main purpose? So the main purpose of identification of features which includes genes with large fold change and high statistical significance between experimental groups. Let's keep it on this way. You have, for example, two groups. One is control, another is treatment. And you want to see what are the distinguished and distinct differentially expressed genes upregulated or downregulated as an output of treatment. And Volcano plot really can help to show your data on a way that's the differences happens in between the genes before and after the treatment. So visualize the magnitude and significance of differences between conditions or classes of samples and used in domains like genomics, transcriptomics, proteomics, metabolomics. A lot of omics studies we use them now. So we are a big fan of this Volcano plot and some other plots like that. So I hope it's basically clear. So if I think like intuitively, what do we think about it? So if we go intuitively, we will see the spots at the top are most statistically significant change, like if you have high upregulated or downregulated. The second is the further from the center horizontally, the larger the fold change. And third, this is the point you should focus. Thus, features at the plot edges are most promising candidates for further investigation. So why I'm saying it that the edges ones are more significant, I will explain through the live demonstration. These are the intuitive things that we expect within the graph. So Volcano plots works with a few mathematical backgrounds. So if you want to first look at, okay, basically we want to see that's what is the basis of these mathematical things. So one of the major mathematical foundation for the Volcano plot that it plots is the p-value calculations. So this is something that we can talk later on, but I would simply say that p-value calculations is one of the way that I will probably make each of these points one video so that you a short video so that you get the elaborate tests on this. So we calculate the p-value using different hypothesis. One of them is null hypothesis, another is an alternative hypothesis. Basically, we will perform the student t-test and we will try to figure out the p-values from there. The calculation could be as simple as is it. Like if it the p-value equal to, you can go to the, like, let me show it a simple thing. Probably one time. That's a different discussion. I don't want to go in there. That will be actually, the video will be much bigger. If someone has real interest, I will be making one by one videos on each of these points and you can have a look from there. So the p-value works for your null hypothesis and alternative hypothesis. If you really want to look at, you can see some few papers by Dudert at 2002, which is where he was using the cDNA microarray experiments to replicate the results. The second part is the log transformation of the result. So you use the results you are finding, you actually do a log transformation of them for better visualizations, and to calculate the fold change fold change in between both of them. So if you want to see the fold change calculation, so if usually, like, if you see two conditions like fold change calculation, so you'd simply do is like probably like condition A divided by condition B will be the fold change calculations in there. So these are the things, those are foundation. You need to have a clear understanding of them. This video is not within the capacity to explain each of them, otherwise it will be kind of a hour-long video, so I couldn't afford that, and people don't watch it actually. That's another issue. And the fourth thing is multiple testing correction. So this is one of the multiple testing correction ideology behind that is like false discovery rate analysis, where you expected the proportion of false positive among rejected hypotheses. So for example, you have a hypothesis, but you have another and all others are rejected hypotheses, and so you try to figure out that what are the false positive in there. So this is there are different methods for that. False discovery rate, or FDR, is most widely used in bulk RNA-Seq and other statistical approaches, and in there we usually follow Benjamin Hochberg procedure, which is most popular in most of the places, but you are free to explore others. So, and the as I am not speaking specifically for the bulk RNA-Seq or other, so I wouldn't go, or single cell RNA-Seq or some other, so I wouldn't specifically go on that. But you are welcome to watch other videos where we have done the world test and t-test in single cell RNA-Seq, world test for bulk RNA-Seq. You can easily get the details. So the last portion in here is that, or the fifth part, is the regression analysis. So regression analysis is kind of a model fitting for multiple variables. So you do the association of multiple variables as by using the linear models. So if you go to the generalized linear models, so you probably do the y equal to beta 0, beta 1, or x1, and like these calculations. You can read certain papers which is relevant to that. So these are the basic foundations for the Volcano plot, which you can go in detailed discussion later on, but now this is the basics. So enough talking. let's go through a demonstration, live demonstration now. That's where we will be creating some Volcano plot using Python, Jupyter Notebook actually in Python, and in R, RS Studio. So to begin with, let's start the Python first. So we go in there, environment, so this is kind of a working directory. I will be going in there. So in here, let's go to IP development, open up IP development. Let's call it the Volcano plot demonstration. So to do and to begin with this, we have to be very careful. The first, let us begin that by working on our specific let's check the recording is okay. Okay, let's begin by importing the different libraries. So one of the basic thing is that what are the common libraries those has been used for data analysis and visualization in Python. So I hope by this time you have already learned details about that. So we have, let's start with importing the libraries. So let's just start with import pandas pd. Pandas is pd. I hope you would know. So pandas is one of the libraries that's used for data manipulation and visualization in there. We use the markdown library. And we import numpy as np. So imported through libraries from the Python. So one of the libraries is that Pandas is used for data manipulation and analysis. Matplotlib is mostly for the plotting library, and numpy is a kind of a array operation and mathematical computation library. So the next part we have is that, so this is the library we have imported. The second part is, so we need to get some data to do the analysis. So let's define the data. Let's try to figure out where do we have some data. Okay, so let's take this one. And you already know how to do the Unix commands, so I'm not going to talk much about this. And then we put it in a data frame, and we use Panda to read the CSV file. Oh, let me show you the CSV file format also in case it helps. So you can see on this CSV file, which is a differentially expressed genes file from bulk RNA-Seq, that gene, and here is the base mean. I'm not sure the visualization, how good is it? Let me make it big up for better understanding. Base mean, and then we have log to fold change. We can add log to fold change, standard value, state, p value, and p adjusted values. So, and these values you can see the p adjusted values, and we can talk some time about the p value versus p adjusted value. And these are the genes in here. So we'll try to pick this up. Sometimes you won't see in DEG if it's generated by DE-seq or bulk RNA-seq, gene won't be written in here. So if you want annotation, you can just manually write it down in here. So let's go back. So we put it in the Pandas data frame and put it like this. So do you see it? So it did, and we check it out actually. Does it synchronize with our the data frame actually we have. So we have it in here. Let me just make it a smaller. So in here we have a print couple of them. That looks fine. So log to fold change, this is one of our major interests, and another one is p-adjustable. You can see details. It's very important you go step by step and go along and understand your data. Now let's calculate the minus locked in p-value. So let's remove it from here. So locked in p-value is minus locked in p-value is not in here. So we need to do a calculation. So we have the DF, which is data frame we have. Then we do negative log p-value, and then minus np log df p-value. So you have the p-value in here. If you want to take the p-value, this is a good option. Or if you want to take, okay, I don't want that, I want to take p-adjusted value. Okay, p-adjusted value, you can take it. There's no problem in there. And this next part is that we initialize a figure. So we PLT is for plotly, if you remember from earlier. So we want plotly to do these things, and figure size. We can go to the figure size equal to 12.8. That will be done, and then we start plotting our data. So if we want, like I want to look at the significant, non-significant, a different level of data, then you can put it on this way. So, but our one of the major interests to look at the upregulated and downregulated data in here. You can always quote the non-significant data, but let's no need to write it up. Plot this data df copy paste this. Otherwise it will take time and use scrutiny in terms of ensuring the data frame is working perfectly. So df and let's keep it as a p-value as we had before. Let's keep it as p-value. df then we have p-adjusted value it define the range of p-adjusted value. For example, we are doing it for upregulated. So let's put it in. Have to write it like this. So regulated So set it up 0.0.05. Change. Then we have df p-adjusted value, p-adjusted value. The same thing. We set it up like for example, we want to set up the 2.5 volt. Then set up TF. Negative p-value. Set it up 5 for example. So these are the basic parameters. So we are setting in here. So let's go through this. Now, what do we have set in here? So we have upregulated genes, and what we have done? We have done the entire setting. I just do it off the line on the record is like it's taking time. So the parameters that we have in here is that plot a scatter, log to fold change, p-adjusted value we have defined, log to fold change, it should what should be the minimum, and then per equivalent or more what at significant, and minus negative log p value. The negative log p value we looked at in here, and the entire thing is set it up in here, so no problem. So the after I set up the this and we run it, you can see it looks like this. So what we will do, we go to the similar setup for the downregulated genes. So we do the similar setup for the downregulated genes. That's in here. So you can see we have defined the different colors in here, red color, and it's in blue color. And then what we are thinking to add up the gene name. So we have set up the certain parameters in here, and we set up the for I. It's a basic gene in weight data frame in plot and data frame log 2 fold change just copied it, log 2 fold change I. That's what the specific gene actually in here ID indicates. So then we go to the just negative state of time. And then I. So data frame we have plot annotation, gene div, okay, this will be the structure in here. So then we'll add the title and labels. So plot title, that's it, Volcano plot, font size 20, plot. We have to fix the label, right? We want to, okay, log 2 fold change, specify the font size, just keep it 15. So whatever, whatever, minus locked in p value. Locked in p value. Font size 15, fine. We need to add up a legend. Plot that legend lock upper right, font size 12, and we set up the plot dot ax h line 5 ls colon split line is adjust to the parameters that's required for the graph. That's all. Two 2.5. Then go to the plot. Show. So AXBH. So the structure has come, and now let's run all of it together. So we take all of it and put them together. So this is the different format we are expecting in here. So now I will run this code together so that you don't have any issue in here. So see, let's run it like this. And now let's restart and run on. So first we imported our pandas as panda, which is a kind of a plot which clearly explains which is a package in Python most commonly used for data manipulation and analysis. Matplot library is for kind of a plotting library, and NumPy is more of for array operation and mathematical manipulation. Then we load our data, and then we bring it into a data frame using Panda, and we have read our CSV file using Panda data frame. We check our data frame: gene, base mean, fold change, log fold change, standard error, statistics, p value, and p adjusted value. And we what if 504 rows and 7 column. So 7 column and total 1504 rows. Then what we do at the very beginning, we define our data set, your data frame we take, and we calculate the simply the minus locked in p value. So we call it like this: calculate minus locked in p value, and then we do just this is next one step is like defining the figure size. Then we look at the upregulated genes, we set it up at p adjusted value how much we want, and we set up like log fold change how much you want, and let me just try to make it a little bit bigger, and negative log p value as a 5 in there. And this is the calculations we have done. We have upregulated genes, we gave it a specific color red. And if you want to see more details on these, and you can see it in here, like so. Next one is downregulated genes. Downregulated genes, and you can go in here. We set up the log to fold change from range, like at what range we want it. So let it begin in here, and then we set up the negative log 10 log p values setup. That's over in here. Then overall we go through the entire setup. The first one is we are setting up the log to fold change and log to fold change range from what to what, like 0.05 to what range in downregulated and negative log p value. And the second one we are equally setting up in different context. So either way, it's upregulated and downregulated in here. I don't want to explain it on such a way that you get confused. So let's go back to the figure. So when we set it up in here, we can actually define this range also. Log to fold change, we have set it up. Okay, this is the range we want, log to fold change and minus log 10 p value. And in the log to fold change, we are going from negative 10 log fold to positive some approximately 9 or 10. We can define these ranges also if we want. Then what we have done, we give a specific color, upregulated as red color, downregulated as a blue color. And the third part is that we wanted to only highlight the genes those are upregulated or downregulated, and we set up the log p value, like log fold change we set up positive and negative. You can see in positive and negative 2.5. So in here we have set it up as a 2.5 log to fold change, and in here negative is 2.5. And then what we done, we looked at the log 10 p value which we have calculated earlier. So to minus log 10 p value when we have calculated and put it in here, and we looked at in positive and negative, we looked at, okay, we set it up as 5. So this is the 5 range, equal or more than 5, those will be only counted as significant and plotted in here. So we set it up as a 5, and whatever above them is counted in here. So this is the basics in here. That's the we have log to fold change 2.5 positive and negative, and log 10 p value that is set up as 5. You can increase the range. Like for example, I want to set it up 10. If we set it up as 10, so then we till here we don't see much. The most important thing is that to look at the upregulated and downregulated genes. So if we take, this is the context of upregulated is, for example, we have two groups. One group is control. Yeah, for example, we have disease like let's take leukemia, and those leukemic cells we have give certain treatment, and we have done the statistics like bulk RNA-Seq, and we have compared the treatment group with the control. Means we want to see the changes within the treatment samples. And to do so, when we have done wall test or the student t-test, the upregulated will indicate the genes those after the treatment was upregulated, and downregulated means the genes those are downregulated after the treatment. For example, if you're expecting that okay, certain genes, for example, we take the KRAS or something like that, those we are expecting to be upregulated. So we see, or in here this context we take CSMD3. So this CSMD3 we are expecting to be upregulated, or FGF7P1. So if we can think from the log to fold change as well as log 10 p value context, this is highly significantly upregulated. And we do it when you downregulate, we are looking for, we are getting RGPD2 from the log to fold change as well as log 10 p value context, that this is highly downregulated. Now, as we promised, the next part of the analysis is reproducing the same data using the R statistics. So let's go. Let's not spend so much time on this. So let's go in the R. Let's clear it up. Start the session. We'll use the same data set to show you that's how we do the same thing in R and Python to generate the same type of graphs. So let's begin with the library importing. So the first thing that is very important in RR is have the beautiful package which is called the ggplot. It actually is a very effective package. So what you have to do is that to analyze your data, the very first thing will be the importing the data package. So we use the library. Let's not type in here, let's type in here. So we take the library and we import the ggplot2. So you have the automatic suggestions and next library and deployer. So deployer package. The next part for us is to import our data. So we'll do the same thing as we have done in Python, but it's a little bit carefully. So to begin with, that we will do first is importing our data. So in there we load our data sets, and this data sets is out of the seq for bulk RNA-Seq. So we will simply just write the importing data is data. That's one data path. Make this object and then put it like this, and what is our data was somewhere in here. Okay, let's copy it. Okay, that's a weird problem. For some reason, are denied to take directly this. So we have our data and we put it in a data frame as we have done in Python. So instead in the Python we have read it as a, if you look at in here in Python we read it on the way that's we read it as a panda data frame, PD for panda. In here we can just the function of read, that will work in here. So we take the data path. Done. Now let's check what happened. Data underscore path. Okay, it's found. I don't know, that could be some error. So we have this. Now we have to calculate the minus locked in p value. So as we have done in Python, we'll use this same data frame but translate it into negative log p value and then set it up like this, minus log 10, and then we take the data frame, we do a calculation and p value. Okay, so we have converted it into calculated the log 10 p value, and then we prepare our data by categorizing the genes based on criteria. So on this portion I just don't want to take so much time. If you want, you just can copy this code and paste it. It is taking too much time. So in here, so data frame we have taken, and within the data frame we indicated the non-significant, and we indicated we can just skip this part also. There's no need of indicating others. Just take the upregulated and downregulated. We set up the same parameters. Like in upregulated, we are setting a negative log value as 5, and downregulated we are setting them as a log value 5, and but the log fold change, if you look at in here, it's in the upregulated we have positive 2.5 and negative minus 2.5. So in the next portion in here is that plotting it using the ggplot2. So to plot it ggplot2, the one of the major function that we are we are going to concentrate is that we um we'll set it up like this. So you want the Volcano plot, and we take our data frame, and yes, this is like a kind of aesthetic. So we are painting the axis will have the log to fold change, and y is if in here we are setting in the negative log pp value, and color type we are setting up. Just put it this way, and geom text we are going through. The text we are going to data frame AS label gene. We want the gene to be annotated as we have done in Python, and we are setting up the different parameters. I don't want that in here. I can just not an insignificant. I don't want it. Leave it. So we want upregulated and downregulated. The upregulated to be red color, downregulated as a blue color, and geom h line we will be setting up just the line, a structural line. That's important, but not so much. So the next point in here, we are setting up the annotated Volcano plot title. So you just set up the title or whatever you want. For log to fold change in the x-axis, and we have the y-axis locked in p-value. We have a classic theme, we don't want much, and we have the legend position, and finally we want to print out the Volcano plot. So this is the basic structure. So now you want to run it. So see, the Volcano plot is generated. To get a better visualization, let me just put it on this way. So our interest was the downregulated genes and upregulated genes, and we set it up the log to fold change as well as locked in p-value. Now the quotient that comes in here, you can set up the axis. That's just something you can do very easily. So let me explain the R code also again, so that you can reproduce it, and you can always welcome to take the code from here, or I will put them in the Kaggle or GitHub. Probably I will just look at the comment box, so I will add up in there. So first we import the library, that's ggplot. That's very important because ggplot does most of the work, the plotting work and a customization feature, and data manipulation part in R is done deployer. So in Python we use the numpy package and pandas and matplotlib. So in here you use the data manipulation, you use the deployer. In there we used the let me see where the code is. Okay, so in here you have used the deployer, and here you are using the numpy package. Then as usual as a standard, we are loading the data, and data frame when we are going, so we are calculating the minus log to p value. Just put it into here. So we have done these calculations now, and then we started doing this specific calculations. They categorize gene based on criteria. This is categorizing gene that upregulated, downregulated. In Python, we have done in here upregulated, downregulated genes. In R, it's always less code and less optimization opportunity also. So we set up the same parameter, and then we started plotting. So if you look at the especially the function that we have used in here is mutate, which talks about the new column creation if we require, and that's column creation based on the case when the log fold change and other changes are occurring in there. So sorry, I'm probably going to do a little bit right side. Okay, you probably didn't see it well, but let me re-explain it. So first we have loaded the ggplot, and in the ggplot we have the ggplot is mostly for the plotting function and customization, and deployer is like a numpy package in Python. It does all the calculations and everything, and then we have the data path, so where we load the data and we read the data, read CSV. In Python we have to use the Panda data frame, and then we do the calculation of minus log 10, and And finally we used the mutate function to create the new column, and when we created the new column in there, and based on these cases, when there are certain conditions, okay, log for change this, and negative log p value is more than 5, we call it upregulated, and the log to full change negative 2.5 and negative log p value for more than 5, we call them downregulated. Then the next part we went is that we can generate the Volcano plot. So we used extensively this ggplot function, and we use the aesthetic mappings, AES for aesthetics in here. Geom point, we are using AES for aesthetics, and we define x-axis is log fold change, y-axis is negative log 10 p value, and we give them specifically geom point and geom text to add point to add up point, and geom text to add up the text. And scale color manual, we get up the color, upregulated is red color and downregulated is blue color. Geom h line, this is for the lines, like how they will be. Then we define some sort of the theme, classic. These are the levels you can always play with, like what you want in the top of the plot and what you don't want. And finally we plot the graph, and we just select it. Let me show it again, as there was some mistake in there, sorry. So you wanted to regenerate, Control L, delete everything, and just click on this, and your data is run. So if you want to go in here, you can see what are the packages loaded, how it is functioning. And then if you want to see the figure, this is the figure in R. And then we have the Python, and we do this, so we can just open in a tab. So if I put them side by side, you can just see the left one is generated using just probably like this, make it a smaller. And this is our package. So we used the same data set, and we generated plot using the R program as well as Python program, and you can see both of the figures in here and here. So this is so reproducible, and you can compare. Okay, link long reading RNA is expressed in here, RGTP2, it's RGPD2 in here. The calculation is a little bit different in R and Python, so that's generated this. And you can look at the CSMD2 top upregulated, CSMD2 top upregulated. So there are certain differences in their structure, but if you can see that's downregulated and upregulated, almost same genes expressed in here and there. So these are the basis of it, and I hope this helps you to understand the Volcano plot generation as well as understanding it, and that's with that the demonstration is finished. If you have any question anywhere, just let me know. And if you want to recap from the very beginning in one line, so it will be something. We started with understanding the what is Volcano plot. It's historically started using in mathematics from around 2000. These are the few observations paper. Then we went first discover rate. Then the importance of a Volcano plot is to look at upregulated and significantly changed upregulated or downregulated genes or proteins or anything we are interested. And then we went through the mathematical foundation, p-value, logarithmic transformation, for change, multiple testing, regression, and then we went for the demonstration. And in the Python demonstration, we have seen we loaded our packages, Pandas, Plotly, and NumPy, and then we loaded our package, we checked our data frame, then we did the conversion, and then we did the upregulated, downregulated, we marked them, annotated the gene, and plotted in here. This looks beautiful. And then we went to the R, and we did the same thing, but a little bit differently. So we went in R, and we started writing, and at the very beginning, we loaded the ggplot2 deployer, loaded our data, and then did the same calculate my logarithm 10, and then We defined upregulated, downregulated. We fixed the Volcano plots, parameters, themed, plotted the Volcano plot. So with that, I will end up today's discussion. Be happy. See you next video.